Hello, everyone. Welcome to Neuroscience. My name is Dr. Michael Weibel, and in today's lecture, we're going to discuss the somatosensory pathways. This includes the posterior column-medial lemniscus pathway, or PCML, which is important for the somatosense of discriminative touch, light touch, pressure, and vibration. The other pathway we will discuss is the anterolateral system. The ALS is important for sensing nociception, temperature, itch, and tickle. Both of these pathways are entirely composed of general somatic afferent fibers, and they are in reference to the body. While we have GSA innervation of the head and neck, that is a different system referred to as the trigeminal system. Both the PCML and ALS are only for the body, and that means they're going to enter the CNS at the level of the spinal cord. All right, well, let's begin with the posterior column medial lemniscus pathway. For the PCML, the body is delineated as upper body and lower body at the level of T6, or the sixth thoracic vertebra. The primary afferent enters the spinal cord via a pseudounipolar neuron, and the soma for this neuron will be located in the posterior or dorsal root ganglia. And in the peripheral nervous system, we refer to this portion of the neuron as a nerve. Let's take a look at a cross-section through the spinal cord. For the sensory pathways, the first neuron in a series of neurons to reach the neocortex is referred to as the first-order neuron. This is also called the primary afferent. For the PCML and ALS, these are both trisynaptic pathways. What that means is that it requires 3 neurons from the innervated target, in this case the skin, to the neocortex. And for the PCML and the ALS, that will be the unimodal primary somatosensory cortex. In both the PCML and ALS, we have a pseudo-unipolar neuron as our first-order primary afferent, and that's very common for general somatic afferents. They usually are a pseudo-unipolar neuron. They're mostly glutamatergic, and for the PCML anyways, the sensory modality is discriminative touch, which again includes touch, light touch, pressure, and vibration. The first-order neuron enters the spinal cord via the fasciculus of Lysauer, also referred to as the posterolateral tract, traverses the posterior gray column, sometimes called the dorsal or posterior horn, and then ascends superiorly along the longitudinal axis of the brainstem to target the medulla. These are then called the ascending tracts. For the upper body, we ascend via the cuneate fasciculus, and we will target the cuneate nucleus. And for the lower body, we will ascend via the gracile fasciculus and target the gracile nucleus. Together, these dorsal columns or posterior columns are sometimes referred to as the posterior funiculus. Once again, the first-order neuron, the primary afferent that is innervating the tissue, in this case the skin, enters the spinal cord via the fasciculus of Lysauer, sometimes called the posterolateral tract, traverses the posterior gray column, and ascends the spinal cord via the cuneate fasciculus for upper body and gracile fasciculus for lower body. Together, these two fasciculi are called the posterior funiculus, or sometimes called the posterior column or dorsal column. Incidentally, if we were below the level of C6, we would only have a gracile fasciculus. So it makes sense that this is medially located because there wouldn't be anything other than the gracile fasciculus. And then as we ascend above the level of T6, we begin to add to the gracile fasciculus laterally, and that becomes called the cuneate fasciculus. Again, the posterior column ascending tracts is composed of the cuneate fasciculus for the upper body, the gracile fasciculus for lower body. The cuneate fasciculus targets the cuneate nucleus in the medulla, and likewise, the gracile fasciculus targets the gracile nucleus, located slightly posterior to the cuneate nucleus in the medulla. From the medulla, second-order neurons decussate across the midline to target the thalamus. The portion of the axon that decussates is referred to as the internal arcuate fibers. These fibers ascend, bound together, in a structure referred to as the medial lemniscus. Lemniscus means ribbon. You could think of a ribbon working its way through a computer, only in this case it's going to connect the medulla and then target the thalamus. In order to enter the neocortex, almost all information must first enter the thalamus. As a matter of fact, the word thalamus means anteroom, the room before the main room. In the case of the PCML and ALS, the subthalamic nuclei is referred to as the ventral posterolateral nucleus of the thalamus. This nucleus has a structure such that the lateral portion of the VPL is for the lower extremity, and then in the middle, the trunk, and then more medially will be the upper extremity. And if we continue to move within the thalamus, the next nucleus that we would encounter would be the ventral posteromedial nucleus of the thalamus, or VPM. And that would process information similar to the PCML and ALS, only it would be for the head and neck. From the thalamus to the neocortex is the third-ordered neuron. These are going to exit the thalamus and enter the internal capsule, which is located between the basal nuclei and the thalamus, and then target the primary somatosensory cortex. For the posterior column medial lemniscus, we have 3 neurons. So this is a trisynaptic pathway, a first-order neuron that innervates the tissue, a second-order neuron that begins at a nucleus within the medulla, targeting the thalamus, and then a third-order neuron that targets S1, the primary somatosensory cortex. The shortest pathway from a target to the neocortex will always be a trisynaptic pathway. Some pathways have many more neurons. They can have 5 neurons, for example. However, with trisynaptic pathways, the second-order neuron is always located within the brainstem, and this is where all of the sensory nuclei are located. Sensory nuclei are small collections of somata that are dedicated to processing raw sense data prior to sending it to the thalamus. The thalamus, on the other hand, is more determined on what you're attending. If you're attending a particular subject, the thalamus will allow information to the neocortex relative to that subject. This means that we'll always have at least a trisynaptic pathway, like the PCML and our next pathway, the ALS. Now, before we move on to the next pathway, I did want to just mention that we could continue from S1, and we wouldn't call it a fourth-order neuron, but S1 sends information to S2, which then is processed for more meaningful understanding. S1 is really just a map of the body, where discriminative touch has entered. What is it a little bit like? Now that we have that information, we'll send that to S2 for further processing, and I'll just note it here. All right, let's move on to the anterolateral system. Recall that the anterolateral system is going to deal with the sense modalities of nociception, temperature, itch, and tickle. I would first like to draw your attention to the fact that we only have the body. We don't have the upper body and the lower body as we did with the posterior column medial lemniscus. For the anterolateral system, we just have the body. And this is a clue as to why the second-order neuron is within the spinal cord itself. Notice here that the first-order neuron is targeting the spinal cord. It's specifically going to fire onto an area within the dorsal horn. Let's take a look at a cross-section of the spinal cord. Again, we have a pseudo-unipolar neuron, which is typical of general fiber types, this being general somatic, but also general visceral afferents. The sense modality is not discriminative touch, but is nociception and temperature, as well as itch and tickle. The first-order neuron will fire onto the second-order neuron in the dorsal horn within an area referred to as the substantia gelatinosa. This is key to how the ALS system functions and why it is structured differently than the PCML pathways. The second-ordered neuron will decussate within the spinal cord via the anterior white commissure of the spinal cord and ascend as the anterolateral system. The anterolateral system follows the same pattern as the ventral posterolateral nucleus of the thalamus, with lower extremity, trunk, and upper extremity following from lateral to medial. And this is because we're going to fire directly into the thalamus. So these are spinothalamic tract neurons. By placing the second-ordered neuron within the substantia gelatinosa, we can utilize a downward analgesia system to stop this synapse so that we can receive or be alerted to potential damage, but then we can stop the continued signal, allowing us to address whatever that potential damage is. That is the key and the reason why we refer to the anterolateral system as a system. But let's go ahead and head up to the thalamus really quickly, and then we'll revisit that analgesia. The spinothalamic tract is structured in the same way as the thalamus. As we said before, the anterolateral system is lateralized so that it's lower extremity, then trunk, and then medially will be upper extremity. Similar to the PCML, the third-ordered neuron of the ALS exits the subthalamic nucleus, entering the internal capsule and ascending to the primary somatosensory cortex. Similar to the PCML, the ALS also integrates at the secondary somatosensory cortex, S2. Unlike the PCML, the ALS also has other targets. I'm going to list a couple of them really quickly. One of these areas is the insular cortex. This is located within the lateral fissure, and incidentally, insula means island, so it's a little island of neocortex. Now, the insula has a lot of different functions, and with regard to pain, it's thought that perhaps the insula plays a role in our discerning the location, the intensity, and the quality of pain, so that we'll have an appropriate emotional response. If you've just got a little paper cut, well, you don't want to have an exaggerated emotional response. However, you want to have some response. Maybe it's the insula that plays a role in adjusting our response to pain. Incidentally, the insula also seems to play a role in empathetic pain where, when we see another person in pain, we're able to share and understand that emotional cognitive aspect of what they are experiencing. So it's a very interesting area of the brain. Still needs a lot of study, but that is one area that processes pain. I'll say one last thing. The insula is thought to play a role in modulating the downward analgesia, which we'll get to in a second. The limbic lobe is extremely fascinating. It is important for our emotional control. It is tied into circuits of the basal nuclei so that we attend to things that we're emotionally involved in. But also, it's part of a circuit that includes the hippocampus that's required for memory formation. And it may be that nociception, as processed by the limbic lobe, helps us form a memory and learn about what has happened. How have we potentially damaged ourselves? Now, this brings me to a final point that nociception is the sense of potential damage that has happened in the first order neuron. Pain, on the other hand, is our subjective experience. So nociception is objective, real-world detection, and pain is subjective, our feeling. We may have nociception without pain, and vice versa. We may have pain with no nociception. Well, speaking of pain, let's take a look at the analgesic pathways that can close down nociception so that we don't experience pain. The reason why we call the anterolateral system a system is specifically because of this downward analgesia, and it begins with collaterals that branch off from the spinothalamic tract and target nuclei of the brainstem. The primary of these is the periaqueductal gray, which is located around, hence the name peri, or surrounding, circling, the cerebral aqueduct. While the internal workings of the periaqueductal gray are a little complex, they work somewhat like the basal nuclei. Ultimately, output from the periaqueductal gray, or PAG, will target the rostral ventral medulla. Within the rostral ventral medulla, there is a subnucleus called the nucleus raphe magnus. We'll not worry about that. But output from the RVM down into the spinal cord releases serotonin, and that serotonin is an inhibitor of the synapse between the primary afferent and the secondary afferent, or the first-order neuron and the second-order neuron. Another target of the spinal thalamic tract is the locus coeruleus. The locus coeruleus, which means blue dot, contains neurons that have norepinephrine or noradrenaline. These adrenergic neurons extend back down into the substantia gelatinosa and release norepinephrine. Similar to serotonin, they will act on receptors that are inhibitory to the synapse, slowing or even stopping the signaling information between the primary afferent and the secondary afferent, or the first-order neuron and the second-order neuron. In this way, the substantia gelatinosa extends throughout the spinal cord and will enter into the brainstem as the trigeminal system. At any point along the spinal cord where information has entered that relates to nociception, that information can be blocked with downward analgesia. That concludes our lecture on the PCML and ALS for the somatosensory pathways of the body. I want to quickly bring up the skin so that we can see what are these targets that we're innervating. To recap, we had the PCML, the posterior column-medial lemniscus, for discriminative touch. That was touch, light touch, pressure, and vibration. Pain and temperature are the primary sense modalities for the anterolateral system, but it also does process itch and tickle. Finally, we do have another set of general somatic afferent fibers referred to as the cerebellar pathways, and these are for proprioception. And then we will be looking at, when we get to the cerebellum, muscle stretch, joint positioning, as well as tendon tension. So we'll see those as sort of the end of the unit occurs. But for now, for PCML and ALS, we have free nerve endings. These are often nociceptors, they may be thermoceptors, and there are some cutaneous mechanoceptors for light touch. The peritracheal free nerve endings are specific to the base of hair, and so if a hair has been touched, these nerves will sense that and send that information. We also have a complex that's referred to as Merkel's disc. This utilizes serotonin, and it's very slow adapting. So it's for more of a static touch. Think if you're holding something in your hand for a long period of time, you'll continue to have that sensation that something is still in your hand. You won't undergo synaptic depression or running out of neurotransmitter. Meissner's corpuscles are for very light touch. These are rapidly adapting. The Parsinian corpuscles are the most sensitive, and they're very rapidly adapting, and therefore they can even respond to very high frequency vibration because they can reset over and over very quickly. Ruffini's end organs are slowly adapting. Again, you could think of holding a cup in your hand, but also the hand position itself. Some tactile information can be combined to give us proprioception. So we can use discriminative touch to also tell us about the location of where our body is in space. And then finally, we have cross end bulbs. These aren't that well understood, but it's thought that they're thermoceptors. And as it stands, we don't really have a very good map for our sense of temperature in the brain. Just kind of fascinating. There's some research that could be done in the future. All right, well, I hope that you found this lecture to be of interest, and I will see you next time. Bye-bye.